Dear students, we are consulting about asexual and sexual reproduction in plants, chapter 1. Last class we have consulted about the vegetative reproduction in plants, both natural methods and artificial methods of vegetative reproduction. Today we are going to study about sexual reproduction in plants. In a plant, the reproductive part is the flowers. Flowers are the most reproductive part of a plant. Flowers are the attractive and colored part of a plant. Flowers are used as a decorative material for all the celebrations. In Tamil literature, the 5 lands are denoted by different flowers. The flags of some countries are embroidered with flowers. The flowers are used in the preparation of the perfumes also. Now we have to see the floral paws. In a flower, there are 4 paws present. They are considered as the floral paws. According to morphologists, a flower is a highly condensed shoot meant for reproduction. As I told you, a flower has 4 whorls, 4 parts. Number 1, calyx. Number 2, corolla. Number 3, androecium. And number 4, gynoecium. Out of these 4 floral parts, calyx and corolla are considered as accessory whorls or non-essential parts of a flower because they are not involved directly in the sexual reproduction. Whereas androecium and gynoecium are considered as the essential parts of a flower because both androecium and gynoecium are directly involved in the sexual reproduction. Androecium is the male reproductive part of a flower and gynoecium is the female reproductive part of a flower. Now, directly you will see the sexual reproduction in plants. The sexual reproduction involves 3 processes. First one, pre-fertilization process. Second one, fertilization process. Third one, post-fertilization process. What is pre-fertilization process? Pre-fertilization process are process taking place before fertilization. What are the process taking place just before the fertilization? They are collectively called pre-fertilization process. Fertilization, you know, it is a union of male and female gamete and form zygote. That process is called fertilization. Post-fertilization. After fertilization, some changes takes place in the flower and they are considered as the post-fertilization process. Now we will see the pre-fertilization process. Pre-fertilization process involves 2 steps. Number 1, development and structure of male reproductive part, that is androecium. Second, development and structure of female reproductive part, that is gynoecium. First you will see the male reproductive part, that is androecium. Androecium is the male reproductive part and it is made up of several subunits. Each subunit is called stamen. Each stamen consists of an anther and a filament. The anther is the fertile part of the stamen because the anther produces pollen grains. The filament is the stalk of the stamen and the anther is the fertile part of the stamen because a number of pollen grains are produced inside the anther. Pollen grains are male gametophyte. What are male gametophyte? A structure which produces male gamete. Structure which produces male gamete is called male gametophyte. Now we'll see the development of anther. Anther is a four-lobed structure. Initially, the anther consists of four-lobed structure. 1, 2, 3, 4. There are four lobes present in each anther. Young anther has a homogeneous mass of cells and that is surrounded by an epidermis. This is the epidermis. The epidermis is the outermost protective layer of the anther which encloses a number of homogeneous cells inside. That is the condition can be seen in the anther primordium. Anther primordium means the young condition of the anther. Then, during the development of anther, in each lobe, a few rows of hypodermal cells become enlarged with prominent nuclei. See, in the next stage, just below the epidermis, cells are differentiated with a prominent nuclei. That is the archisporium. The cells are called archisporial cells. In the archisporial cells, cell divisions takes place. Periclinal division and anticlinal division takes place. Periclinal division means parallel to the epidermis. So the first division is periclinal division. Due to the periclinal division, two types of cells are produced: primary parietal cell and primary sporogenesis. Primary parietal cells are produced outer to the archisporial cell. Primary sporogenous cells are produced inner to the archisporial cells. The primary parietal cells undergo a series of periclinal and anticlinal division and form 2 to 5 layers of anther walls. So the primary parietal cells only gives rise to the anther wall. There are 4 types of anther wall. The anther wall consists of 4 layers. One is epidermis, just below the epidermis endothecium, just below the endothecium middle layer, and the innermost layer is tapetum. So the wall of anther consists of 4 layers: outermost epidermis, innermost tapetum, in between epidermis and tapetum endothecium, and middle layers can be seen. So that is about the development of anther. That's all today's topic.